Hello AP Bio Bruins, and this is Mrs. Farouk, and I'm going to talk about passive transport. And when we talk about passive transport, we're basically going to talk about transport in a cell without using energy, and that's basically diffusion, facilitated diffusion, and osmosis. And when we talk about these three types of diffusion, we always relate back to how does the structure of the cell membrane allows things to move across the cell membrane. Now, under passive transport does not require energy. So the net movement of molecules is always from high concentration of molecules to low concentration. So if you look at this picture here, on this side of the membrane, there's a lot more molecules. So it will randomly move towards from high concentration to low concentration until it reaches dynamic equilibrium. And when we say dynamic equilibrium, it means it's still moving, but it's moving in equal rates. So that's where the word dynamic comes in. And equilibrium meaning in equal rates. So now in this scenario, you have solutes both in equal amounts in both sides of the membrane. And where in this side, you have great amounts of one type of solutes on one side, and another side you have another type of solutes. And so both of them are independently moving from high concentration to low concentration. Now, under passive transport, you have two types of transports. You've got diffusion, which again is the random movement of molecules moving from high concentration to low concentration. And facilitated diffusion is the movement of, again, molecules from high concentration to low concentration, but this time you have the help of a protein. And this protein basically can act like a gate or a pore that facilitates the diffusion of particles. And usually these particles are large or charged that would not normally pass through the membrane. And if you remember, membrane is made up of phospholipids, and these phospholipids are permeable to lipids or nonpolar or small particles. So anything like water or charged particles will need help or facilitate by a protein. Now, water diffuses across a membrane, and we call that osmosis, but water moves across what's called an aquaporin. And basically it's a protein that acts like a pore that only allows water to pass through. And what's so neat about the aquaporin, that along its sides it has a polarity that attracts this water molecule to pass through. And in fact, this water by one molecule at a time would pass through this pore, and it's called an aquaporin. Now, here's some terms that you need to know to understand cellular transport. So the word, again, solution and concentration. So what is a solution? So a solution would contain two things. That's solutes in a solvent. And how we measure solutes are moles over liters of water. Now, many of you know chemistry. That's a weird W. Water. We call this molarity, and a symbol for molarity is M. Okay. Now, the terms you probably heard, solution is hypertonic, hypotonic, or isotonic. These prefixes, hyper means more, hypo means less, iso means equal, and these all are referring to the concentration of solutes. Okay, concentration of solutes in water. Oops. So if you have a high concentration of solutes in water, that means you have less water and more solutes. In hypotonic, you have less solutes and more water. In isotonic, you have equal amounts in solutes and water. Now, we also describe besides hypertonic, hypotonic, isotonic, the word water potential. And when we talk about water potential, we're talking about the concentration of water from one area to another. So basically it's in a symbol called psi. And think of Poseidon. It's what do you call it? Like a Poseidon's. So we call that psi. And water potential, think about it as a potential water movement from one area to another. So if you have something that has high water potential, we're talking about it that it has more water and less solutes. And something with less water potential would have less water and more solutes. So we're talking about comparing two different areas and movement of water, and we refer to this as a concentration gradient, now in this case in terms of water. Okay, now the water potential is measured by this formula. Water potential equals the water potential due to pressure plus water potential due to solutes. So if you have pressure, that adds to the water potential. We're basically talking about physical pressure. And when you have water potential due to solutes, that decreases water potential. So that's always negative. And it's negative because the more free water molecules you have, the more it will do work. But when you add solutes, the free water is tied up. So here, for example, you have water, and some of the water is attracted to another solute. So now you have less free water molecules, and that's why you have a negative sign there. Okay, so how do you calculate water potential and predict the direction of water movements? Again, we go back to this key formula. Water potential equals water potential, the pressure potential plus the pressure of solute potential. Now, pressure can be both positive and negative. It's usually positive. In plant cells, you'll have pressure exerting. Usually in animal cells, since there is no cell wall, the pressure potential is 0. And in open air, like in a beaker, it would be 0. So if you look in this picture here, this represents positive pressure, right? So therefore it's 0.23. And then you have solute potential. Notice it's negative because it decreases the water potential as you have more solutes, because it decreases the availability of free water molecules. And so any solutes decreases the number of free water. So here, if you see in a picture, you have solutes, and here it has a negative 0.23. And then when you add these two, in this case, it's a 0 water potential. Now, if you want to figure out the solute potential, this is the formula you would use, where I equals ionization constant. So for example, if you have a sodium chloride solution, sodium chloride, when it's in water, breaks up into 2 ions, so its I would be 2. And then if you have sugar, that does not break up in water, so its I, the I would equal 1. Now C equals the molar concentration, and that's usually given in a problem. And R is the pressure constant, which is right here, 0.0831 liter bars per mole Kelvin. And the temperature is in Kelvin, and remember, if you're given in Celsius, you just add 273. So calculating water potential, predicting the direction, again, if you have a plant cell, that cell wall again exerts the pressure inside the cell is being exerted and limited by the cell wall. So here you have to determine, okay, this cell is in a solution, in this case a 0.4 molar sucrose solution, so it has a negative 0.9, and then the potential here is, from pressure is 0. So what would happen if you place the cell in a isotonic environment? In an isotonic environment, this plant cell would be flaccid. Now, if you put it in a hypertonic environment and water potential in the cell is more in the cell versus outside the cell, then the water will leave the cell. Okay, and that will lead to plasmolysis. Okay, what plants like is a turgid and conditions. And turgid conditions is when the plant cell is in hypotonic environment, and where water will enter the cell, and then it won't burst because of the cell wall, and we say it's turgid. So water osmosis always from high water potential to low water potential, or in other words, in hypotonic solution to a hypertonic solution. So if you look at this U tube, and we call it U tube because it's in a U shape, and notice these dotted lines, this is a semi-permeable membrane, and this membrane will allow certain things to go through. So in this case, solutes are not going to go through, but it's allowing water to go through. So if you compare A to B, A has more solutes, so this represents solutes. So we say A is hypertonic because it has more solutes and less water, and B is hypotonic because it has more water but less solutes. So it has less solutes, and this has higher amount of solutes. And so because it's higher solutes, you have less free water, and this has more free water. So the water B has a higher water potential versus A has less water potential. So the water is going to move from hypotonic to hypertonic, right? And now if the solutes are going to move, the solutes will move until it reaches equilibrium. So but in this case, water will move in that direction. In this scenario, we have equal number of solutes and equal number of amount of water. So we say this is an isotonic. So this is isotonic and B is isotonic. So the movement of water leaving and entering are equal. So again, water moves from high water potential to low water potential, or from hypo to hyper. So whenever we use the terms hypertonic or hypotonic or water potential, we're always comparing two different environments, and we compare one side to another side and to determine which direction the water would go. And again, we're looking at the amount of solutes, or we're looking at pressure, which all determine the amount of water. So again, water is moving from high concentration of water to low concentration of water across a semi-permeable membrane. And that's what makes osmosis, because osmosis is the movement of water and you need a membrane. Okay, so looking at this picture again, predict and explain the movement of water. Okay, so in the first one, which direction would the water move? So A we would describe as being what? Hypotonic, where B is hypertonic. So the direction would water go from, it would go A to B, because you have more water in A than B. Now in this case, you have B being hypotonic, oops, or we could say it has a higher water potential, and A is hypertonic,